as to how ribosomes and protein synthesis work together to form proteins. So a reminder that we are working through the central dogma of biology, which therefore means that DNA is transcribed into RNA, which is then translated into proteins. Proteins are used by the body. They give you your traits. They build things such as the pigment in your eye, so you have blue eyes. So RNA will code for specific amino acids. The amino acids will then build our proteins. So we're going to do that now. So just a kind of intro to translation. So we have been going over in the, last week, we said that transcription happens when mRNA is formed from our DNA. So that happens inside of our nucleus. But now our mRNA has come outside of our nucleus, full of all of those letters, all of that code that we got from the DNA. So now we're going to actually use translation to process sequence of mRNA codons. So what's a codon? So a codon is a set of 3 letters that come together to tell a tRNA, transfer RNA, which amino acid needs to be built. So a codon, example AUG, AAC, or UCU. But how do we know what these letters mean? How do we, basically, these are in a foreign language right now. Our body, this is not helpful to our body until we start building amino acids. So that's what this fun table is for. So codons, we have different sets of 3 letters. So let's continue to look at AUG right here. So we would go to this giant table, which gives us 64 possible bases. You have 4 different letters that you can start with. You can either start with a G, U, A, or C. And then for the next letter, you can also only have a U, C, A, or G. Same thing here. And then for that last one. So let's start here. We want to see what UUA builds. Well, that builds leucine. Or CUU also builds leucine. So there's a couple different possible ways to build different amino acids. So this whole outer level is our amino acids. So let's look again. So to read the codons, you start in the middle circle with the first letter of the codon and then move outwards. So if our first letter is a C, we would start and find a C. Our second letter is an A. We would go find in this row, find an A. And our last letter is a C. That builds the amino acid histidine. Okay, CAC is histidine. Methionine is a special one. We need to remember this because that is AUG, which is our start codon. So whenever tRNA or transfer RNA sees AUG, they can start building our amino acids. We also have things that are called stop codons. That tells us to stop building something. Okay? So how does our cell read the genetic code? Well, our cell reads the genetic code by taking sets of 3 nucleotides, 3 different letters, and it codes for a specific amino acid or a specific stop, which means to stop building something. Okay? So again, translation is where we are outside of the nucleus. We take sets of 3 letters from our mRNA, and our tRNA will start building. This is our tRNA. Our tRNA will start bringing over different amino acids that match up to our letters here. Okay, so our letters tell us which amino acid we are building. Well, our amino acids are actually going to form together. They're going to connect to each other, and that will actually build our protein. So again, proteins are used to kind of share our traits or show our traits. So our polypeptide connects them all together. And then we have this chain right here, which would be our protein, a bunch of amino acids connected to each other. Okay, our tRNA can stop building because we got to our stop codon right here. This blue blob is also known as our ribosome, and that basically just helps bind the two together, your mRNA to your tRNA. So how do ribosomes use mRNA and tRNA to assemble